Hey guys, welcome to my YouTube channel Knowledge of Biology with Anjubute. I hope you all are good. Today I'm discussing with you what is the gene cloning process. So we know that gene is a small fragments of DNA, and cloning means our specific targeted sequence we are transfer one bacterial body into another. So look at that. What is the gene cloning process? Gene cloning. So guys, very important question. Why we are used to be this technique? Because this technique are responsible to be produced as a insulin gene, and insulin genes are responsible to be produce protein. Insulin protein means they are synthesized by insulin's protein, and this insulins we are use many drugs or therapies also, especially diabetes patient, because diabetes patients bodies are not contain proper amount of the insulins, and that's reason their body's glucose are continuously circulate into blood. It is very dangerous and very important to be this glucose are absorbed. So that's reason we are used to this technique to be produce insulin proteins to be they are absorbed as a more than glucose. Okay, so look at that. So in this technique guys, firstly take by double standard DNA. For example, this is our double standard eukaryotic DNA. Okay, so in this DNA, we want to be make small fragments. Okay, so take by one experimental tube, and in this tube add by this double standard DNA plus restriction endonuclease enzyme. We know that guys, restriction endonuclease enzymes are responsible to be make small fragments. Okay, and we want to be our specific targeted sequence. They are cut specific of the base pairs, and we get specific sequences. So and imagine to be makes fragments, and then next take by second experimental tube. In this tube we add vector molecule plus DNA ligase enzyme. Vector molecule used as a vehicle to be transfer specific targeted sequence one bacterial body into another. Okay, and DNA ligase is responsible to be joined specific targeted sequence into the vector molecule, because we are at first experimental tube into the our vector molecule with restriction enzyme, so they are provide specific cut. For example, it's our vector molecule, and they are specifically cut to be these sequences. Okay, then this vector molecule we are add also second experimental tube with DNA ligase enzyme, and imagine to be they are combine each other, and we get this recombinant DNA. Okay, and this recombinant DNA we next process is this recombinant DNA we are add by host cell, for example E. coli. That is our host cell. Before add to be this recombinant DNA into host cell, we are provide shock treatment in this cell. Why? Because very important to be their cell wall membrane are loose in short period, and easily our recombinant DNA add by this host cell. Okay, they already have bacterial chromosomal. Then two important points to be this recombinant DNA, they are self replicated and have selective markers. What is selective markers? They are antibiotic resistant gene. means they have capacity to be produce also antibiotics. Okay, for example ampicillin and tetracycline also. Okay, next process, um, after replication we are isolated our recombinant DNA. Okay, so then take agar plate. Agar plate contain more than nutrients. It is very important to be replication of the recombinant DNA and produce more than colonies plus antibiotics agar. Okay, at specific antibiotics, for example ampicillin. Okay, for example imagine to this our recombinant DNA are produce ampicillins. Okay, so also had to be agar and antibiotics both are. Then after some time we observe more than colonies of this plate, and this is not a normal, it's our recombinant genes. Okay, this is a process to be isolation of the specific targeted sequence replication and protein synthesis, especially insulin. Okay, I hope guys you understand to be this video. Take by screenshot, and thank you so much.